Dear Professor Bosch, let's start with the very beginning. You are a speaker of a collaborative research center. So what does this actually mean concerning the microbiome or the meta-organismic research?
Our collaborative research center, Origin and Function of Metaorganisms, is focused on the function of the commensal, of the stable microbes in many different host model systems, including plants, worms, flies, humans, hydras, and probably some others. So we try to compare the interaction between microbes and their given host or plant animals and hosts to understand the basic mechanisms behind. Each system will have its unique language and rules. But our hope is that each system also will reveal fundamental and evolutionary conserved mechanisms. And by analyzing them in a comparative manner, we hope to get to the basic languages which evolved to maintain a stable, healthy holobiont. We hope that by this comparative approach, we also, and we have included medical scholars in our collaborative research center, we hope that we also can inform the medical community about what comes out here, and then it's up to the medical community to use this knowledge or not to use the knowledge. This collaborative research center integrates sciences from the computational sciences, the mathematical and the metabolic modeling. We are collaborating with the Max Planck Institute in Plön on that. It integrates botanists, zoologists, microbiologists, so many different disciplines. And one challenge, but also a vision of future natural sciences is that we have to give up borders between these distinct disciplines. There is no single institute or even department anymore which will find the fundamental rules by itself. We need the collaboration with our colleagues from different disciplines to get, with this enormous amount of data, with this complexity of mechanisms involved, to get to a deep understanding of something so simple as a simple freshwater polyp.
You were working with Hydra as a model organism already, as I understood you right, but your connection to the microbiome research happened more or less by accident. Can you tell us how this works?
Yeah, it was not an accident, but it was a great chance. A young graduate student came in the lab, and I had many mutants, different species of Hydra in the lab, studying them on a developmental biology background. And we came to this idea that for his thesis, it might be interesting to study the microbiome, the bacteria which are associated with these animals. Sebastian Fraune is his name, and so Sebastian sequenced the different species of Hydra, and at one point came to me and said they all have a different microbiome, they all have different composition of bacteria. And I thought a little bit about it, and I said that's impossible because these animals are with me for 20 or more years, living in plastic containers, getting artificial water, artificial food, traveling around the world. How can they have different bacteria? I don't believe you. Go out in the wild and collect new wild animals and sequence them again and try if you can repeat that, what we have seen in the lab. And at that time, I was relatively new in North Germany, and I did not even know if these animals are existing in the wild. But Sebastian went out in the Plöner lakes and collected some of them, did immediate sequencing, and observed that more or less the microbiome of the animals from the wild is very, very similar to the microbiome of animals which have kept for 20 years in plastic dishes. So then we came to the conclusion, wow, that's interesting, that's very stable. And because it's so stable, it means that there must be selection on that, which you hardly can disturb. And there must be a reason for that. Ever since then, we came to the conclusion that obviously bacteria are a major part of complex life forms. And in the meantime, many different labs with many different organisms have confirmed that finding, including work by Howard Ochman, who has shown in several papers that the microbiome of humans, of Homo sapiens, is distinctively different from the microbiome of the closest ancestors, which is gorillas or orangutans or chimpanzees.
If this microbiome is so specific, there must be, as you said, a selection. So who or how is the organism, the host, shaping its microbiome?
Yeah, that's an important and interesting question. I think everything starts with novel technologies. I mean, this first paper from us came out in 2007. That was the beginning of a high-throughput sequencing possibility. We couldn't have done this research 20 years ago. But with this new technology, some invisible important components of life became visible. And they also changed many dogmas and changed our view on many things. In answering your question, it particularly changed the views on the role of the immune system. If you think that every organism, plants, animals, humans, have a distinct microbiome, your question is, how is that maintained? The innate immune system and the adaptive immune system. Simple organisms have only the innate immune system, which consists of antimicrobial peptides, of toll-like receptors, and of a conserved signal transduction pathway. In addition, vertebrates have an adaptive immune system with all the T cells and the antibody variation, etc. And when thinking, how can this stable bacteria community vertebrates, the adaptive immune system may contribute to maintain the more complex microbiome. That the immune system is there to defend pathogens and to fight pathogens. We had here in Kiel a collaborative research center on antimicrobial peptides on epithelial defense. And the strong belief was, for example, antimicrobial peptides, as a major part of the innate immune system, kill bacteria. And we were looking for them, we were isolating them. Some of them were killing multi-resistant Staphylococcus aureus, so we had patented some of them, always in this belief that the immune system is the defense system. After finding the microbiome as a stable partner and looking for mechanisms, my view completely changed. And I propose, and I have written that the function of the immune system is not so much to fight pathogens, but to maintain the holobiont. And of course, we use antimicrobial peptides, and they are killing microbes. But on the same side, they also maintain a stable composition of the microbiome by keeping the abundance, for example, of certain bacteria in a given quantity. And many years ago, my colleague Margaret McFall-Nye has published a similar view on the adaptive immune system, where she proposed that nobody really knows what is the function of the adaptive immune system because many animals live without it, and very successfully. Actually, 95% of all animals do not have an adaptive immune system, but are very successful here for 560 million years. So Margaret proposed in a very important paper that in more complex vertebrates, the adaptive immune system may contribute to maintain the more complex microbiome. Today we know that T cells, T cell responses, and the signals which are activate T cells, all of that is in very close interaction with the microbiome. And so yes, it seems to be true that both the adaptive and the innate immune system are the mechanism which the animal and the humans use to maintain that stable microbiome. So we use our immunity to maintain this complex being together of different organisms.
So if the host invests so much in recruiting its microbiota, there must be a lot of advantages. There must be some important meaning of this thing. And are there costs on the other side? So what is the meaning for, you said already it's against pathogens, but are there costs as well?
Yes, there are tremendous costs, and I refer to my colleague Martin Blaser, who realized and who published very early on a very influential book, what happens if microbes are missing in our body? And the answer is we are all very sick and suffering on many different diseases, which are mostly chronic diseases, which are, as the WHO organization calls, non-communicable diseases, because they cannot be transferred from one person to the other. They are there probably because of our lifestyle, and in particular, and that's what Martin Blaser points to, because of the ubiquitous presence of antibiotics, which are not only there because we take too many antibiotics in our daily life, but we uptake antibiotics with our food. Antibiotics are everywhere. And what they do is they reduce the diversity of the microbiome in us as well as in agriculture animals and fish. And by that we disturb the microbiome, the healthy microbiome. And the consequences are enormous. The consequences are all the modern diseases which more and more people suffer from. Chronic inflammatory diseases, adipositas, allergies, asthma, neurological disorders. Autism is thought of having to do something with that. And this is an increasing number. It started in the Western world. It's now all over the world, also in developing countries. It seems that the modern lifestyle is contributing to that. But economically, for nations, that's an enormous cost. So we better try to understand what's going on, what are the reasons, how is this complex multi-organismic nature of life organized and regulated, and we may even think of, can we extract fundamental rules, but also maybe can we prevent this type of developing diseases by understanding and by maybe exploiting beneficial microbes so that we return to a more healthy state.
Do you think, looking 10 years in the future, that there will be, that this microbiome research will lead to a real practice in medicine, and when you go to a doctor, that the microbiome will be an important part of your anamnesis?
I'm absolutely convinced. But I also warn, I think at the present day, we are in the early days. Scientists, researchers, and also companies get first insights in the importance of these microbiomes, but that's far from a very general application yet in a daily clinic. But I can easily see that in the future you go to the doctor, and the doctor is not so much interested in taking a blood sample, but he takes your feces and he gets the feces sequence, and he will make a and from the sequences you will see what kind of intestinal bacteria you will have. And I just got my microbiome sequenced by a colleague who asked me for that, and I have a written printout now of my microbiome, and this microbiome tells me what I'm eating and maybe what I should change to return to a better health status. And I think this type of diagnostic, but also the knowledge of important microbial-derived products, which may influence our body function, logically should lead into novel therapeutics. I mean, you can go today in many supermarkets and you get probiotic yogurts and whatever, and you go to the pharmacy and probably the person behind the counter will sell you something. I'm very skeptical about that, if that is really that what we mean. And because, I mean, what we really aim to is to get a mechanistic, a functional, a fundamental understanding of the mechanisms behind, of the pathways behind, and also of the detailed products. And I could envision a research, host-microbe research, which aims at the isolation of these metabolites, of the synthesis of these metabolites, of the critical evaluation of these metabolites in particular pathways, and then a rigorous testing in vivo model systems before going to a clinic, and then applying it to some microbe deficit disorders, as many of these chronic inflammatory diseases are really called. And that process probably will take at least the next 10 years. I do not see a single product today on the market which fulfills this criteria. There is also a lot of hype in the field. The microbiome is getting very, very sexy. Many people try to jump on the train by describing microbes and specific bacteria compositions in all kind of different contexts. That per se is really not very interesting anymore, because that microbes are there, and maybe they're also specific in certain places in our body or on plants. That's already very well known. The question of the future is, what are they doing? The functional analysis, the interdependence, the way of communication. How do hosts and microbes communicate with each other? How do microbes communicate with each other in a way that they still maintain a stable community in our body? How does the host interfere with that communication? And what happens if this communication is disturbed, for example, by our way of living, by medication, by certain health products, or by food or something like this? So there is lots of research still needed, requiring an interdisciplinary field. Only by that, this complex new world, which was invisible for so long time, and only became visible because we have destroyed it, becomes something which we can understand and maybe even use for our purposes.
Mm-hmm. So we made the way from the very primitive organism like Hydra to humans, to medicine, and probably medicine is the area where microbiome research or metaorganismic research will be important for everybody of us. But I would like to go one step back and look at biology. So if you view this field from a biology point of view, what is the most important message for you in this way?
The most important question in biology is, which was asked by Schrödinger, what is life? I think biologists, natural scientists want to understand the basic principles of life. Now comes another technological revolution. And as I said, the revolution made visible the previously invisible world. And what I personally think now is that life is multi-organismic. Complex processes of life, including development, including interaction with the environment, including the immune system, including the interaction of the nervous system with the environment, they all are multi-organismic. We talk with the environment, which is microbes in the environment, but also other organisms. And the challenge of the future will be to incorporate that multi-organismic interactions in maybe understanding fundamental rules, how life is really made up of, and what are the major building blocks. We hypothesize that multi-organismic interactions in the evolution of life are older than multicellularity. They go back to unicellular organisms, like paramecium, like simple ciliates. They all have symbionts, they all have bacteria. So probably major mechanisms of living cells, they never happened in a sterile petri dish. They always were in exchange with the environment. And microbes were first. Microbes were there first. All multicellular life originated on a biofilm. So it's not so astonishing that some aspects, at least, of signals which cells need to make a certain decision derive from the microbial environment. Coming back to the question, what is life? I think we cannot disentangle that from the realization that all gets together with everything. And microbes were there first, 3 billion years. Multicellularly evolved around 460, 440 million years on a biofilm. So we better should take into account when we study developmental, immunological, cellular mechanisms, that all these mechanisms are tightly connected with the environment. And when we disturb the environment, these host cells get sick. That's an exciting time to live in.
I think it's a very nice take-home message, so thank you very much.
Thank you very much, Kegel. Thank you.